So, let's follow the path of endosymbiosis and get these steps in your notes just to make sure you have them, because this always appears on the AP exam. Okay, now in step 1, so we're down here now on the left side of this diagram. In step 1, we can see the evolution of our ancestral prokaryote, more than likely a real primitive bacteria right down here. So we're going, this way time-wise. So here we're going to start with our ancestral bacteria. You see here just some scattered DNA, didn't do a whole lot. In step 2, this primitive prokaryote begins to evolve, and over time new characteristics appear, expanding its genome and requiring a nucleus to house the larger genome. So now it starts to develop an endomembrane system. All right, that endomembrane system then gives access to the outside of the cell from the nucleus. So now here you see a nuclear envelope forming here. So we've gone from a prokaryote that has no nucleus. Now over time it's gotten bigger. You can see it's gotten bigger. And it does more. So therefore its genome has expanded, probably now has chromosomes. And those chromosomes need to have a nucleus in order to house them. So now in step 3, you have a full-blown endomembrane system where materials can travel in and out of the cell into the cytosol and into the nucleus. You now have a full-blown organelle called the nucleus that houses now the DNA that's now fully developed in the cell. In step 4A, label it as 4A, we see this cell engulfing the aerobic heterotrophic. All right, so we're down here engulfing the aerobic. Aerobic means oxygen, it uses oxygen. Aerobic heterotrophic prokaryote. Now remember, heterotrophs don't make their own food. They've got to seek their food elsewhere. So in order for this mitochondria to produce energy, it's got to find organic molecules outside the cell. So this now cell has engulfed this aerobic heterotrophic bacteria, presumably because it doesn't do as good a job of manufacturing the energy on its own, and this bacteria's job is to produce energy through respiration, cellular respiration. So it engulfs it. So that's 4A now, engulfs this aerobic heterotrophic prokaryote. Now this was the evolution of the very first heterotrophic eukaryotic cell. Let's look at that. Heterotrophic because it's got to get its food somewhere else, and eukaryotic because it now has a true nucleus. Remember, this is a prokaryote because there is no defined nucleus. This now is a eukaryote because it has this defined nucleus that houses the DNA. And now it also has engulfed this aerobic heterotrophic prokaryote. Now, in step 4B, so label this as 4B, this heterotrophic cell now engulfs a photosynthetic prokaryote, a photosynthetic. So remember what photosynthesis is the ability for an organism to convert light, photo means light, to convert light into glucose and energy. So now it's engulfed it's a bacteria that's very good at producing its own food and also produces ATP as well. Not nearly as efficiently as aerobic respiration, but still produces energy. This then evolved into the first photosynthetic eukaryote autotroph. Remember that autotrophs now are organisms, plants in particular, that can make their own food and energy. So these are now autotrophs, so they can do two things. They make their own organic molecules, and they can use those molecules to manufacture their own energy. That's extremely important for you to understand, because what happens now is you have an evolutionary split. You have the ancestral heterotrophic eukaryotes down here that become the first animal cells because they don't have chloroplasts. So it goes now off on its own little evolutionary split. And then over here, you see the formation of the first plant cells. Because plant cells, remember, have plastids. This is a chloroplast. And they have mitochondria. So now, from engulfing two separate kinds of bacteria, these cells have now taken a different evolutionary path, and now the animal cells will develop out of this heterotrophic eukaryote, and our plants will evolve out of this photosynthetic prokaryote. So now we have a working theory of how both plant and animal cells evolve. Now remember, it's just a theory. Nobody really knows that this has actually happened. It's a theory. See it right here. Theory is unproven, but it has a bunch of data to back it up. So, and that involves the engulfing of other bacteria that's specialized in respiration and photosynthesis. And that this theory is referred to as endosymbiosis, first developed by who? Lynn Margulis. Okay. This is an excellent opportunity to review the cycling that photosynthesis and respiration are involved in. Remember, whenever you think of photosynthesis, you think of chloroplasts. Whenever you think of respiration now, you think of the mitochondria. Okay? So what I want you to do is write these equations in your notes on top of each other. One is a word equation and one as a chemical equation, and make sure they're balanced. So go ahead and do that in your notes right now. Now, let's get some observations in your notes. Observation 1 that we've mentioned earlier is that the cycling of breathable oxygen and carbon dioxide happens because of these two chemical equations. You see now up here in photosynthesis that glucose, C6H12O6, is surely formed, but also O2, breathable oxygen, is formed. So now, in photosynthesis, plants give off breathable oxygen into the atmosphere. Then down here in respiration, we're using that same glucose molecule and the oxygen in the atmosphere, okay? And what's given off now is carbon dioxide to the atmosphere that plants can now use, some water, and here's our developing energy molecule, ATP. So the cycling of oxygen and carbon dioxide now are what we want to see in this situation. And also the production of ATP as an energy source for both kinds of cells. Okay, so here you see another visual example of the cycling we're talking about. The carbohydrates produced by plants are used by them for energy, but is now available for animals to consume and get energy from through the digestion process. If you're more of a visual learner, this might be a diagram you'd want to have in your notes. It's fairly simple, and who knows, you may see this diagram on the test. I have no idea. But if you're more of a visual person like I am, this might be a much better way to look at it than our stacking up the equations like we just did. If that's true, have at it. Now, let's end our tour of the cell by reviewing some more key organelles contained in plant and animal cells. We're not going to look at them all, but we're going to look at the key ones. And we've already looked at mitochondria and chloroplasts, okay? And we've already mentioned food and water vacuoles and their functions, along with lysosomes like you see here. So vacuoles are another example of important organelles, as are lysosomes. So You already have that in your notes. We're not going to waste any more time on it, but we're mentioning the important organelles here. Now, vacuoles have a particularly important function in plant cells. Write in your notes that vacuoles have 5 things that they do, or 5 important functions in plants. Let's get them in your notes. Number 1, vacuoles stockpile, you know what the word stockpile, they save or store up, proteins or inorganic ions for the cell. Number 2, they also deposit or dispose of metabolic wastes. Number 3, they store plant pigments. Number 4, they store defensive compounds to use against herbivores. That's true. Herbivores stay away from plants that have a bad taste, or in some cases, poison in them. And 5, vacuoles function as a selective membrane to control what comes in or out of a cell. So those 5 important functions in vacuoles in plants. So make sure you get those down. Okay, now peroxisomes. Write that peroxisomes store additional digestive enzymes for the cell and are contained in both plants and animals. And that these enzymes break down 2 things. They break down fatty acids into sugars. Also write that they work to help detoxify the cell by removing alcohols and other poisons. This process produces hydrogen peroxide, which then is broken down into water and oxygen in the peroxisome. All right, that does it for our quick tour of the cell and review of the most important organelles in each type of cell. And my goodness, by now you should be able to tell the difference between an animal cell and a plant cell, and you should be able to differentiate the organisms that one has that the other doesn't. And you should also be able to explain the theory behind how these cells actually developed, the theory of endosymbiosis. So again, it's a theory, but it's the best working theory that we have now that has evidence behind it. Okay, that's it for our tour of the cell part 3. Take a picture of those notes, send them to Moodle, grab the study guide, answer the questions, and we'll see you in the next series of podcasts on respiration and photosynthesis. Bye.